Thank you very much for that fantastic introduction, Billy. I appreciate it. And thank you all for having me here today. So it's my pleasure to talk about today symbioses or potential symbioses involving the world's largest bacteria. And I want to point out that this is work that's been done together with, oops, sorry, the joint lead investigator on the project, Dr. Beverly Flood, and also my PhD student, Dalton Leprich, who's done a lot of the fantastic imaging that you'll see in this presentation. Okay, so what are the world's largest bacteria? Diamargarita namibiensis is kind of the canonical giant sulfur bacterium, and these are called Diamargarita namibiensis because that means sulfur pearl of Namibia, and that's where they were first discovered. And you can see that they have this appearance of, they look kind of like a string of pearls, right? And so that's where they get their name, and they were originally described and published on by Heide Schultz off of Namibia here. And so just like the Humboldt upwelling system that we're studying off of Namibia, you have the Benguela upwelling system. And it's sort of an analogous in many ways in terms of the oceanography and the biogeochemical cycling, and also many of the organisms that you find are similar in both places, but with some interesting differences. So Diamargarita is the largest known bacterium with individual cells getting up to 1 millimeter in diameter. So here's a fruit fly for scale from the original, Heide's original publication with individual Diamargarita cells here. So you can see just how large they are. And also more recently, these very interesting Diamargarita cells published on by Valand et al. That are sort of like a septate filament here that's not, you can see there's like a division septum here, and this one is kind of divided off. And you can see kind of nascent little septa here, but these are centimeter long chains that you could argue are one, and they did argue are one cell, right? Or you could say that it's sort of in the process of dividing off of these daughter cells. And we see that, we've seen that previously in other morphotypes of Diamargarita, where you have kind of the budding off of daughter cells. But in this case, these are extremely long cells / filaments, right? And these organisms are so large because we think because they store lots and lots of storage compounds. And so what you're seeing here actually is a relatively thin layer of cytoplasm that's surrounding a larger aqueous vacuole. And that aqueous vacuole contains high concentrations of nitrate, which they use as a terminal electron acceptor for their chemolitho-autotrophic oxidation of sulfide and other reduced sulfur compounds. So what do I mean by that? So just as kind of like a simplified cartoon schematic of lithotrophy or sulfide oxidation with oxygen, if you think of our respiratory chain, we have an electron donor, which is organic matter, and it's passed through the electron transport chain, and we pump protons, and we convert that proton gradient, that charge gradient, into ATP, and then we use that ATP to drive all of our various cell processes, right? And similarly, you could think of, in the case of lithotrophs, chemolithotrophs, as using something other than organic matter as the donor of those electrons. In this case, hydrogen sulfide or other electron donors could also be H2, which Diamargarita has the genetic potential for that as well. But really, this is kind of a simplification of what the sulfur oxidizers are capable of, and there are lots of different pathways, complex pathways that that carry out a variety of different transformations of sulfur compounds that can have a variety of different redox states. So it's actually quite complex, and there's still lots to learn. And this is based on models of sulfur bacteria that are in culture, but Thiomargarita we don't have in culture yet, despite trying for years. So as I said, these organisms are, you could kind of think of them as like a nitrate scuba tank in a sense, in that they have this really large volume here, but a lot of the cytoplasm is kind of this relatively thin layer, and a lot of the cell volume is this aqueous vacuole. And this vacuole contains nitrate that they use as kind of a reserve of electron acceptor. And these organisms also store lots of other compounds as well, things like polyphosphate. So polyphosphate is this polymer of phosphates that's made by lots of different organisms, and in some cases stored to very high concentrations as an energy reserve, or oftentimes as a stress response. And they store other molecules as well. They store elemental sulfur, they store glycogen, and so on. So these organisms are really, in a sense, adapted to storing various different metabolites. And that's most likely because they live in an environment where they have changing conditions, changing access. Over time, they have changing access to either the electron acceptor or the electron donor. And oftentimes when we think about microbes and try to grow microbes, we think about providing continuous conditions that are kind of static, as opposed to in natural systems, including the habitat where they live, where these organisms live, conditions change over time. Indeed, organisms like their close relatives, Beggiatoa and Thioploca, actually need access to both an electron donor and electron acceptor, which they find at kind of gradient interfaces in the sediment. And so these motile organisms, Beggiatoa and Thioploca, they basically move up and down to find that redox interface, or in the case of Thioploca, they can take up nitrate closer to the surface of the sediments, store it in the vacuole, and then move down into the sediments deeper where there's hydrogen sulfide, their electron donor available. And so they're using their motility to kind of transit between the electron donor and the electron acceptor. As far as we know, Thiomargarita are not motile. It was originally posited that it may be something like a gas release that moves them up into the water column where they have access to electron acceptors. But I think, having studied them for quite a while, I think what they're doing is that they're just transiting redox space by waiting it out over time. So they're just, conditions will change around them at the sediment water interface, and so if they can store what they need, they can just wait for the conditions to change around them, which they do seasonally because you have changes in upwelling intensity and therefore changes in biological oxygen demand and nutrient and organic matter delivery to the sediments. Okay, so this family, the Beggiatoaceae, are members of the gamma proteobacteria, and they include Thiomargarita, Thioploca, and Beggiatoa, and others as well that are not as well talked about, like Thiopolula, which is like the little sulfur pill, which is like mini Thiomargarita. There are a whole bunch of different phylotypes and morphotypes. And you can see here, as Thiomargarita, there are a couple of different major species, if you will, or phylotypes, the Namibiensis type, and then the Nelsoniae type. And then you also have here Mari Beggiatoa, and somewhere here's Thioploca. And so they're all quite closely related here, the marine Beggiatoaceae. And then there are also, as well, freshwater Beggiatoaceae, Beggiatoa and Thioploca in freshwater systems, but they're very small and they don't have this vacuole, or they're smaller, I should say. And you can find actually these organisms, these giant sulfur oxidizing bacteria in a number of environments. And actually, when I was a postdoc, I went to certain environments with the idea that I could find them if I found the right kind of conditions, and if I had the right image, search image in my head. And indeed, I was able to find them at a number of places where they hadn't been reported before, by just looking and having the idea of what I was looking for. And that actually, I think, is an important point to consider when you're thinking about kind of your career and the tools that you use, and our kind of increasing reliance on molecular approaches, and in a way kind of abandoning a lot of the microscopy, right? A lot of these organisms here we wouldn't actually have discovered using molecular approaches, because there are very strong biases against the amplification of these organisms. And in some cases, you can take samples that are full of Beggiatoa or Thiomargarita visually and get kind of no amplification unless you're using very clade-specific primers. And there's a number of reasons for that I can talk about later. But again, it underscores the importance of using a microscope still and looking at what there is out there in nature, and not just taking a sequence-based approach, which is of course important as well, but both are important, I think, depending on your question. So here is how one of my kind of first foray into potential symbioses with Thiomargarita. I had an opportunity to go down on the submersible Alvin to 2,000-meter water depths off of Costa Rica. And so here you have subduction of a seamount, and basically you have the subduction of the Cocos plate here. And as a result, you have lots of kind of fluid escape going on. So you have methane and hydrogen sulfide that's basically venting out along kind of fracture surfaces along this subducting seamount. And so all along the kind of the rock outcropping surface at various different fracture interfaces, you can see the enrichment of mats of sulfur oxidizing bacteria. So you can see here there's lots of mats of bacteria. I didn't show any pictures of it, but there's also clusters of animals that have lithotrophic symbionts like Bathymodiolus. So you'll be going along, and maybe there's a mat here of sulfur bacteria, and then maybe there's a little pod of Bathymodiolus mussels here. And here's kind of a cool picture. So these are gastropods, snails that are deep marine, that are eating these mats of sulfur oxidizing bacteria. And we collected some of these snails and observed something very interesting. So first we saw that there are actually like kind of individual Thiomargarita cells. They're not chains, but they're individual cells that are kind of around. You can find them around here. But then when we looked at the snails themselves, what we saw is this kind of almost like if you know what chia pets are, it looks like a chia pet of Thiomargarita on the outside of these snails. And so these are all these structures here. Those are individual Thiomargarita cells with daughter cells at the end that are budding off, right? And this basically, what we think it represents is sort of like a life cycle that if you're familiar with Colobacter, which is kind of a model organism for dimorphic cell cycle in normal-sized bacteria, it looks a lot like that, where you have these smaller individual cells, spherical cells that are, we think, kind of more the dispersal stage, and then those attach to a surface, and then they get longer and longer, and then they start to bud off these kind of daughter cells into the environment. And there's lots of different morphotypes and interesting morphologies associated with this. In some cases, these spherical daughter cell type forms will attach and grow longer like this, but in other cases, they will actually undergo reductive cell division, making something that looks very similar to an animal embryo, actually. And so you can see here these snails, these living snails, are covered with dense accumulations of these Thiomargarita. And we observed both in their positioning when we collected them, and also when we brought them back to the lab and kind of watched their behavior in tanks of sulfitic sediments, that they actually turn over and expose their bacteria to the sulfidic sediments for a while, and then they go up to more oxygenated environments. And then they kind of make this trip back and forth. And they're really exposing their kind of soft parts when they like flip upside down to bury these bacteria in the sulfide, which is kind of an interesting behavior. It may be that they're acting as a platform for moving these bacteria between the place where they have electron donor, the sulfide, and where they have the electron acceptor, oxygen or nitrate. And they may be getting the benefit of having a food source. There's lots of patches that, like this one, where you can see it looks like the Thiomargarita have been grazed off by some either other gastropods, or there's also a very small limpets that live on these shells that are grazing as well. So it looks like there's some kind of very interesting trophic interactions that are going on here. So this was kind of my first foray into thinking about Thiomargarita and its involvement in potential symbioses. And so I started really getting interested in the physiology of these organisms and trying to grow them. We tried to grow them for quite a long time, and Beverly, for example, is really, she's very talented at growing sulfur oxidizing bacteria and has isolated a number of them. But even using all of our different approaches that we've tried, we still have not been able to grow Thiomargarita in the lab. So we turned to doing other approaches like using redox sensitive dyes and so on to kind of look at the metabolism that was going on with these organisms in hopes that we could figure out the conditions that they like best. You can see here those dyes actually stain Thiomargarita, but then you can also see that there's oftentimes a sheath of polysaccharide. Sometimes it's very thin or almost non-existent. In other cases, it's quite thick, that contains lots and lots of other bacteria. And these are the obvious kind of big filaments, but there's lots and lots of other cells here. And you can see that here with some of Haida's images as well. Lots of bacteria are attached to the outside of these bacteria. And so we started thinking of them also not just as Thiomargarita as a epibiont on some animal, but Thiomargarita as having its own microbiome. So it has all sorts of bacteria that are attached to it. And so we started looking, going to Namibia and seeing if we could see if there are any kind of specific associations between Thiomargarita or any of these other sulfur bacteria that you find off of Namibia, and particular bacteria that are attached to them. So epibionts on the big bacteria. And so we basically took an approach of collecting sediment samples at various different localities, and then we would pick out the giant sulfur bacteria and then do amplicon sequencing on the bacteria that we picked out, and then also compare it with the sediments to see if there was something that was enriched in the bacteria that we were pulling out relative to the background kind of sediments. And so we just basically took kind of an amplicon approach. I won't explain, the details of how that works, but our results basically showed that there were, in some cases, some groups like the planktomycetes that did show kind of an enrichment for particular types of Beggiatoiaceae, including the Thiomargarita type here. So you can see here the planktomycetes are kind of more better represented with these in association with these non-filamentous Beggiatoa relative to the background sediments. And kind of the major take-home points for the amplicon work with the epibionts is really that there are about 30 or so ASVs that were kind of enriched, and those include the planktomycetes, some other organoheterotrophs, lots of nanoarchaea. We have no idea what these nanoarchaea are doing, but they were present in a lot of our Thiomargarita samples. And also some sulfate-reducing organisms and also some sulfur oxidizers as well. And a lot of these organisms that we're finding are heterotrophs, and a lot of them are probably getting something by breaking down the sheath material, right? So there's this EPS kind of sheath. They're probably living off of that in one form or another. So we think that that sheath material probably enriches for a lot of these organoheterotrophs. And it could also be that you have things like sulfur cycling that are happening on the scale of these giant sulfur oxidizers and sulfate reducing bacteria that are living on their surface. Usually sulfur cycling is separated kind of spatially, but it's possible in these kinds of associations where you have giant sulfur bacteria and sulfate reducing bacteria on the outside that you have kind of very tightly spatially coupled sulfur oxidation and sulfate reduction. Which could lead to some interesting isotope signatures and so on. We've thought about that before. Okay, so this is basically kind of where we were interested in the bacteria that live on the outside of Thiomargarita. And we were also spending a lot of time looking at Thiomargarita under the microscope and trying to figure out how they grow and what they're doing. And we made an interesting discovery, and that is that here's Thiomargarita under the microscope, and you can see there's lots of inclusions here, lots of like inclusion bodies here. A lot of that's sulfur granules and what have you, polyphosphate and other things. But if you look, you can see there's this kind of like brownish reddish cloud here, and if you look closely here, you can see there's like all these little specks, right? And if you look carefully under the microscope, you can see that those things are moving around. And I noticed this in a few cells, and then I started to see it in more and more and more, and I started to see it in a lot of them. And some of them had lots and lots of these little structures that were moving around in there. And I thought, wow, that's really interesting. I wonder what those are. Could they be cells in there? And so we had a bunch of different hypotheses initially. We thought maybe these were bacteria that were just kind of breaking down dead Thiomargarita, because in some cases it's hard to tell if your Thiomargarita cell is dead or alive. And so it could be they're just dead and some bacteria got in there and breaking them down. Or it could be that maybe they have some kind of giant viruses that infect them. Or maybe these are actually some kind of very small dispersal stage or something like a spore or something, right? So we had a bunch of different hypotheses, but at first I was the only one in the lab who thought maybe these are bacteria that are endosymbionts. And other people were like, no. But I held out. I was like, I think they could be. And so then we started to do some more work, including some electron microscopy. Let me step back and say there aren't actually hardly any examples of bacteria that have endosymbionts living within them. And so there's a well-studied example of mealybugs. So these are insects that have bacteria living inside of some of their cells, and then bacteria living inside of those bacteria. So it's like Russian dolls nested, right? But they're within the eukaryote host cell. They're not free living. There is actually this interesting paper in the Journal of Microscopy Research and Technique by Larkin and Henk back in 1996, where they were looking at some Beggiatoa in TEM view, and they saw structures that they thought might be some kind of endobiont. And so, but this paper was kind of overlooked, and we weren't aware of it for quite a long time. But yeah, they basically showed that there were some structures in there that resembled endobiont bacteria. And we then looked at our cells with TEM, and sure enough, these things look like bacteria to us. So there are lots and lots of these endobiont bacteria, some of which have inclusion bodies in them. Here, you can see that. And you can see ones that look like maybe they recently divided, and lots of kind of similarities in terms of their morphotype. And I should step back and say the scale of this is really kind of strange, because the Thiomargarita cells are so big, the TEM only captures like a small little patch of it, right? And so this is like the Thiomargarita would be like, arching at this scale, it would be huge, right? So this is just one little edge of it, and this is the outside, and then this is the vacuole, and this is the Thiomargarita cytoplasm, and these are all the endobionts. And then we had to address the question of whether or not these these organisms are actually alive, metabolically active, and also the host. Is the host metabolically active? Oh, I forgot. Also, we did some additional work using cryotomography to also try to show this relationship. And the reason for that is that, Thiomargarita is very difficult to study using conventional kind of electron microscopy techniques because it's basically like a water balloon. If you add a lot of these fixatives and dehydrating agents, it tends to cause damage to the cell ultrastructure. And so then we had to also, during the pandemic, try to get some cryo-imaging done as well that would try to keep the cell more intact and allow us to kind of look at these spatial relationships in greater detail. But also we needed to look at these organisms and see if they're actually metabolically active, both the endobionts and the host. And so you can see here in this is a confocal microscope slice through the Thiomargarita cell. So this is the Thiomargarita cell, and here are the endobionts. And this is a kind of a typical backlight live dead stain. And then we also use these tetrazolium redox dyes to show kind of active metabolism in the Thiomargarita cells. And we also used amino acid labels. So here, these are amino acid labels that are taken up by both the cells outside in the sheath and by Thiomargarita and the endobionts. So you can see them moving around in there, and so they're taking up amino acids. And also you see apparent inheritance of these kind of clouds of endobionts across the division plane here, right? So even though we can't necessarily follow this over time, it seems clear that these clouds of endobionts here are mirrored across this division plane, which suggests that this cell is actively metabolizing and dividing with these and potentially transmitting these endobionts to the offspring or the daughter cells. So who are these endobionts? So that was a challenging question to get out as well because there's all these bacteria on the outside, right? And so we basically had to extract those endobionts from the inside in order to get figure out, sequence them and figure out who they are because we have to keep them separate from all the bacteria that are on the outside. And so we have a number of different approaches for that, including like the device that you use for in vitro fertilization under the microscope that you can use to extract the endobionts. But really just like smashing them open and the endobionts will come out, and then you can like grab them up and do PCR on them and what have you. So Nicole talked earlier today about the tube worms, right, at the hydrothermal vents. So these are the seboglinid tube worms, and they have bacterial symbionts. And interestingly enough, our endobionts are very closely related to them. So here are the seboglinid endobionts here of the tube worms. Here, a couple of different types of tube worms. And then here are our Thiomargarita associated endobionts. So it actually, their relationship or their phylogenetic placement makes sense that they're endobionts. It supports the idea that they're endobionts, I should say. So then we wanted to go back in and make sure using kind of the full cycle rRNA approach so we can sequence something, but then we can design a FISH probe to go back and confirm that that those are what we think they are. And so we designed FISH probes and went back in and then looked at those endobionts. So we used FISH probes to look into Thiomargarita, which is again challenging, but we see like enrichments of the endobionts with this FISH probe beneath the surface of Thiomargarita. Some pockets here of enrichment of those endobiont cells. And then we also did metagenomics on these organisms. And interestingly enough, the endobionts have very similar genomes to their host. And you might think, well, that's odd. And it is kind of odd, right? They have, just like the Thiomargarita, they have the genetic potential for denitrification, for sulfur oxidation, for carbon fixation using the Calvin cycle. So very similar to Thiomargarita in many ways. But then something Thiomargarita doesn't have is these endobionts have the genetic potential for a number of different secretion systems, which in other organisms, including pathogens, are used to target eukaryotes for eukaryotic cells for infection and various other things, right? So you can, bacteria use them in a number of different ways, not just infection, but that's one possibility, right? One thing that the secretion systems can be used for. So one hypothesis, and we don't know if this is right, but it's possible that these organisms are, in a sense, stealing the nitrate from Thiomargarita, or at least not stealing, but getting inside, right? So if you imagine Thiomargarita, there's a selective pressure for Thiomargarita to store nitrate, because then if it runs out of electron acceptor around it, it has a storage of it, right? But if you're another organism that can't store this, a small bacterium, but you still need nitrate, or you can use nitrate as an electron acceptor, if you can get inside this scuba tank of electron acceptor, then you can make a living there, right, during these times when there's nothing to breathe. So that's a hypothesis. I don't know if it's right. There's certainly, there's other ideas as well, but that's just one idea that we're kicking around for why you might have an organism that's so similar in terms of its genetic potential to Thiomargarita living inside of it. But still, there are a lot of open questions. How are the endobionts getting into Thiomargarita? Are the endobionts interacting with them? If so, how? Thiomargarita is known to be involved in lots of different types of biogeochemical processing. So they're important for sulfur oxidation, for transformations of nitrogen, phosphorus. They're thought to be involved in making phosphorite deposits off of Namibia. So they're thought to be really important in terms of their geobiology and their biogeochemistry. And we don't know if these endobionts are involved in that in any way. And also, is there some evolutionary significance to this Thiomargarita endobiont relationship? And what I mean by that is that endosymbiotic theory posits that the acquisition of a symbiont was really important for eukaryotic evolution. You had the acquisition of the predecessor to the mitochondria and the chloroplast that really is thought to have driven a lot of the evolution of the early eukaryotic cell. And there are lots of examples in the modern biosphere of bacteria that live inside of eukaryotic cells, right? But there are really not that many examples of bacteria living inside of other bacteria. And you would think maybe there would be more. Bacteria living as infecting eukaryotic cells, or being engulfed by eukaryotic cells, or living in symbiosis with eukaryotic cells, but not many examples of prokaryote endosymbiosis, which you'd expect that there would be a number of them or more of them, given this kind of idea of how the endosymbiotic theory is thought to have been really pivotal for early eukaryotic evolution. And part of that is because we think of the bacterial cell wall, or at least one way we explain that, kind of canonically, is that the bacterial cell wall is not very dynamic, and it's just this like shell, and it doesn't do stuff like phagocytosis, right? That's kind of the image we have in our minds. But I think we're starting to appreciate that maybe it's not that kind of clear cut. And we have some evidence in Thiomargarita that suggests that the ultrastructure can be quite dynamic. So if you recall, I was talking about amino acid labeling, right? So amino acids can occur as both D and L chiral configurations. And so D amino acids are discriminated against by the ribosome's translation machinery, so you don't get them in proteins, right? But where you do get D amino acids is in peptidoglycan, right? So peptidoglycan in the bacterial cell wall is found in the peptides that kind of cross-link the glycan strands. And so here, like D-alanine, you can find in the peptidoglycan. And a number of researchers have, starting with this paper, Crew et al. In 2012, have used fluorescently labeled D-amino acids in order to track the synthesis of new peptidoglycan. And so you can see some examples here of peptidoglycan synthesis along the division septum in E. coli and some other in Streptomyces, some kind of pulse experiments here where you can see the kind of incorporation of these D-amino acids into peptidoglycan. And so we started using these to see as another kind of tracker to look at these cells that oftentimes you can't tell if they're actually metabolically active. And so we started using them to kind of look to see, is Thiomargarita making new peptidoglycan? And sure enough, you can see similar things in Thiomargarita where they have enrichment of D-amino acids along the division septum and so on. But what we didn't expect is that we also found that there are these very interesting vesicle-like features that were actually incorporating these D-amino acids. And so you can see here's a confocal slice in multiple different dimensions, orientations of the same stack, 3D stack. And so this is a dividing Thiomargarita cell. Here's the division septum. And you can see all these structures here are actually incorporating these D-amino acids, which suggests that they're peptidoglycan-bound vesicles. And here you can see what looks like the cell wall is actually infolding here. And so we think that the cell wall is folding in and basically making these vesicle-like features, which vesicle-like features are well known in bacteria, but they're not known to contain peptidoglycan. They're usually membrane material. And they usually bleb outward, not inward, and they're way, way smaller than these. So these can be, we're talking like many microns, tens of microns, very large features, right? And so they seem to actually, in some way, translocate into this central vacuole, and they start from the ultrastructure out here. And we've shown using other peptidoglycan-specific dyes that these are not just some artifacts of the D-amino acids. Other things like wheat germ agglutinin and even electron microscopy supports this, that these are basically some kind of delamination or infolding of the cell ultrastructure to make these vesicle-like features. And sometimes they include vesicles in them, like sulfur granules, and sometimes they don't. And if you look closely at these endobiont images too, you can see that there's electron-dense kind of vesicle-like features here, and then around and many, in fact, of these endobionts, you can see these kind of electron-dense features, and we can actually see that with light microscopy too. We see that there's, in some cases, the vesicles have concentrations of endobionts within them. In other cases, the endobionts seem to be distinct from the vesicles and not found in them. So how are these vesicles forming? Interestingly, Thiomargarita's genome has actin homologs. They also have a number of other interesting characteristics to their cytoskeletal proteins that are kind of different from a lot of bacteria. These organisms have thousands of copies of their genome. They have introns in their in their 16S gene. They have introns in functional genes. In a lot of ways, they're not exactly like our canonical bacteria. In some ways, they're kind of eukaryotic-like, or they have features that are sort of like eukaryotes. And so maybe this endobiont internalization is somehow associated with vesicle formation. And we're kind of pursuing that hypothesis right now with infection challenge experiments and additional imaging and so on. We don't know exactly, we're not necessarily saying for sure that we think it's phagocytizing these. It could be that maybe those bacteria are exploiting some kind of weakness and are able to get into the cell along those planes. But we're still investigating that, but it's quite a challenging set of experiments to figure out. Okay, so lots of new things to figure out, and I just want to thank the funding agencies, NSF, the Simons Foundation, the Sloan Foundation, and also the course, the RGNO Namibia course, which is a course that Kurt Hanselman started that was modeled off of this course in Namibia, and this is where I collected a lot of the samples for the work that I showed here. And those samples were collected with the help of the Ministry of Fisheries in Namibia and the crew of the Mirabilis, which is their research vessel in Namibia. And so I want to thank you very much for your attention.
That was awesome. Yeah, please, we have time for 2 or 3 quick questions, please.
I'm gonna try to be quick. A ver, what do you think that the symbionts are giving to the Theo Margarita? So the bacteria let those symbionts and seems that outwit the host defense against these symbionts.
Yeah, maybe they don't. Maybe they can't defend themselves. That's what I think. I think that I don't think that they have any way of defending themselves. That's the only thing I can think of, that they don't actually have some kind of immune system or immune response. I don't know that, but that's just the thing that comes to my head, is that they can't prevent it. And I should point out though that not all the Thiomargarita have them. Some of them have very dense accumulations of the endobionts, and some of them have a very small amount, and some apparently have none. Initially, we thought that a lot of them didn't have any, but now if we look closer and more carefully, a lot of them have a very small number, and maybe about 20%, or depends on the site, have a lot. Yeah, and so some appear to be heavily infected, we call them, but others just kind of have a few or lightly infected. So don't know. Yeah.
Well, thanks for your presentation, Professor. I think that I have a quick question. Why the article related to the flood 2021 compared the Theo Margarita with its associated taxa, but only in a phyla level, and instead of the same level of Theo Margarita genus? I don't understand why the analysis just consider a general phyla level or a general taxa level with the Theo Margarita, which is a specific level.
Right. Well, we were just looking at the major groups that are associated with the Thiomargarita, and we were kind of just plotting those major groups. But those are all ASVs, so there's specific. You can look at the taxonomy at, all the way to the genus and species level, but we just plotted the enrichment by kind of the general grouping to look at what general groups are associated with Thiomargarita, broadly speaking. Yeah, but the data is there, and you can pick it apart down to whatever level you want. All right. Well.
Do you find them, the endosymbionts in
Here?
No. When you did the association, you didn't know that you had.
Oh, yeah.
Now when you look back at your data, are they there?
Yeah. They're there. Yeah.
Could I?
Sure.
Okay.
Okay. I had a question about because you watched this membrane approach to study. So it's these endosymbionts are mainly associated with the membrane, or are they like in the center of the cell also?
Yeah, we find them in both. We find them in the cytoplasm, and then we find them apparently in the vacuole type region. And in some cases we have not as commonly, but we have actually seen them along the division septum, even on the outside. And so they have been enriched there on the outside, just along the division plane, but that's only a few instances where we've seen that. Usually they're not like, oops, sorry. They're usually not just like hanging out on the outside. Yeah, yeah. It's usually, yeah, they're inside.
Okay. Also, a quick question. There's still, I understand your hypothesis that you think they're in competition to the Thiomargarita. Have you looked, for example, for different carbon fixation pathways that anything beyond Robisco?
No, they both have Rubisco. Oh,
And there's
actually,
no-
Thiomargarita has Rubisco and RTCA, but the endobionts have only
That's all.
Rubisco. Yeah.
And so there's none of, for example, the use of an incomplete CO2 fixation like the 3-hydroxy-4-propionate bicycle, where they would take up acetate and propionate from nothing. Any indication for heterotrophy in the symbiont?
I don't
I mean,
believe
in
so.
the endo, whatever you want to call
Yeah,
it.
yeah, yeah. I don't believe so. No. But
Endobiont, that's neutral.
In Thiomargarita, yes. And Thiomargarita can use a lot of other things. Thiomargarita can use H2 potentially and other and
So they're
heterotrophy.
strict obligate autotrophs based on their metagenomes.
Yeah,
the endobiont.
Yeah, the endobionts are, but not
In
Thiomargarita.
contrast to Thiomargarita.
Yeah, that's right.
So it's really only competition. The other, can I one real quick? Okay, sorry, because you said only 2 or 3. Okay, okay. Is that actin and tubulin homologs are common in bacteria? It's not just how should I say it? My impression was that you thought this might be something specific to the endosymbiotic relationship, but many bacteria have these homologs. I just wanted to
But
understand where you were going with that.
Yeah, I didn't show any pictures of it, but there's actually like filamentous type structures, and these are different from like the canonical actin homologs like MAMK, yeah, they're a different homolog, and they're arranged more like a structure, a large scale kind of structure in the cell. I guess MAMK is associated with magnetosome formation in magnetotactic bacteria. So that's an actin homolog. And that does make it makes these vesicle-like structures. But here you have the vesicle-like structures, and this is just something we observed within the last few months. Vesicle-like structures have these long kind of like cables coming off of them that actually will label with actin-specific labels. Yeah. Okay.
We've used actin labeling and it labels to so much bacteria. It's not
Okay. Yeah. I'd like to talk about it more.
Okay, thank you very much, Jake.